Hello, in this video we will discuss about the what is gene and one gene one polypeptide. According to the scientist which that is proposed this hypothesis. So the Archibald and Garrett basically is provide the what is gene one polypeptide, which that is the basic and strong concept we should need to understand. So the Archibald and Garrett is Archibald Garrett and William Bateson basically is the two scientists which concluded in 1902 about that they concluded that the certain diseases among their patients were more prevalent in particular families by examining several generations of these families. So important thing is that disease cause due to the recessive allele of the same homologous chromosome. So the homologous chromosome means the one half is come from the parent mother and half is come from the father. So the basically is important thing is that the chromosome basically is the I am trying to understand basically as well. So the chromosome contain alleles. Basically allele means the companion to each other. So it means the one diploid zygote containing our body contain in the form of two a pair of the chromosome. This is a chromatin pair, and that is half is come from the mother and half is come from the father. So here, so that is contain allele. Allele basically is the gene which that is the basically mask or express the another allele of the parental genome. So here is basically is the A and small a, which that is the dominant, for example insulin producing gene, and allele and non-insulin on the mask non-insulin producing gene allele. So here is the very important disease according to the Archibald Garrett and William Bateson, which that is the alkaptonuria is the disease, which that is lead to here is the expression basically is the normally. So the dominant A allele is dominant and mask the another allele effect. So our urine contain a homogenetic acid. This is converted into the simpler substance by the activation of the enzyme. So it means our urine will not produce the black color due to the breakdown of this acid. So the non-black normal gene function. So the non-black if normal gene function. While on the other hand the recessive gene is basically, for example, the A allele will be recessive. It will not produce the product. For example, the black urine production, and homogenetic acid will not. Homogenetic acid will not break down. So it means oxidized to produce black color. So that is the important things which that is related to the gene, and that gene is in the form of allele, and one allele which masks the effect to another allele, that is called the dominant. If the masked allele is known as the recessive allele. So that is the simple way to understand. While on the other hand, the from Garrett's finding, it could be interested that the information encoded within the DNA of chromosome acts to specify particular enzyme. So the particular enzyme are present which that is used for it according to the Garrett's. So that means here we will discuss about the what is the enzyme and what is the polypeptide used for it, and one gene one polypeptide we will going to understand. So let's begin to understand. First is the basic concept for getting the knowledge to understand the one gene one polypeptide hypothesis. So here is the basically I am drawing the DNA structure. DNA is in the form of chromosome. The chromosome contain a histone protein. Basically 60% histone protein, while the 40% is the DNA present in the chromosome. So this DNA is the histone protein. 4 type of histone protein contain a 8 subunit of histone proteins known as nucleosome. and DNA is wrapping around the nucleosome, and this DNA is basically is made up of antiparallel containing a nitrogenous bases and ribose sugar and phosphate group by a phosphodiester bond. So here is the adenine and guanine is the double ring is known as the purine, while the thymine and guanine and uracil is the pyrimidine, which that is the single ring. So during the mutation, this mutation can be point mutation for the deletion of the nitrogenous base adenine, for example, or addition of the nitrogen base, for example the guanine or adenine. And on the other hand, the inversion, that is the mutation, and this mutation is lead to change the environment inside of the body of the organism. So here is the 1941, the series of experiment by a Stanford University through a George Beadle and Edward Tatum. According to the George Beadle and Edward Tatum, is the Beadle and Tatum deliberately set out to create a Mendelian mutation in chromosome and then study the effect of these mutation on organisms. So the Beadle and Tatum's procedure for isolating nutritional mutants in Neurospora we will discuss also. So according to the Beadle and Tatum, that's produce the that is give the complete evidence of the Mendelian trait. Mendel was saying that the gene are present in our body, which that is lead to generation after generation dominant and recessive on the basis of opportunity. So that means that Mendel says that two alleles segregate during gamete formation, for example sperm or egg, and fertilize at fertilization randomly during the fertilization, the unite randomly during the fertilization. So that means produce the new trait by a dominant or recessive. So let's begin to understand on the other hand the basically the mutation. That mutation basically can be point mutation, but this point mutation will lead to whole changes and not grow the minimal media, for example. So this is the normal wild type neurospora containing test tube, and minimal medium we will providing. This minimal medium the neurospora can grow easily, while this minimal medium contain a sucrose, vitamin and water just, and not complete amino acid. So this neurospora can grow, but after the growth of neurospora we will provide another. The George Beadle and Edward Tatum will provide the X-rays of ultraviolet light because they know about that the X-ray can mutate the DNA of the neurospora. So after the mutation, the asexual spore or ascospore of fungi Neurospora bread mold or fungi will be produced after he grow the growth on complete medium amino acid containing basically, and this complete medium will provide the meiosis after the meiosis to produce that product of one meiosis in the neurospora and select a one of the spore. So first we will select the one and we will confirm that the complete medium is able to grow even mutated neurospora. While the product of one meiosis, this basically is the select one of the spore. So after the selection of the one spore, we will provide the complete medium first and with amino acid 21. It means the complete amino acid containing medium is basically is important for the growth even the mutated neurospora also. But important thing is that it's contain a 21 type of amino acid. So that means all amino acid are present in the nutrients. So neurospora is not required to produce amino acids such for a growth. So that means in the minimal medium the neurospora will not grow. Why? Because the minimal medium contain not complete amino acid, while the sucrose and vitamin and water for the wild type neurospora can grow, but the mutated neurospora can't grow. So that means the non-growth factor is basically the amino acid are not present in the minimal medium, while the complete medium contain a 21 type of amino acid. So that means we are confirming here the amino acid is important for the mutated neurospora growth. So here is the we are getting a minimal medium to give the neurospora each tube containing pyridoxine, amino, benzoic acid, choline, inositol, nucleic acid, folic acid, arginine, and niacin, riboflavin, and thymine, and another. So that means the arginine containing minimal media is growing, but another medium is going to rest. So that means another medium is not growing. Why? Because the arginine is most required for the growth, but the arginine is not producing from the neurospora. Why? Due to the mutation of the gene. So the gene is controlling the production of arginine. How it's possible? Let's begin to understand. Here is a neurospora. This neurospora is basically is normally neurospora will produce the arginine. So arginine is produced. So the mechanism of the production of arginine we will discuss. So here is the haploid ascospore. This haploid ascospore contains a chromosome. This chromosome will not going into the crossing over. So that means we are choosing the neurospora for understanding the better in the research. So that means the neurospora will not lead to mutation itself, but we will providing the mutation. So here is a basically nucleosome is basically the DNA wrapping around the nucleosome, and here is the transcription. We will discuss basically here is the DNA antiparallel which contain a nitrogenous bases. So that means it contain a gene segments. So this DNA contain a genes. This gene is the specific nitrogenous basis. On the basis of the specific nitrogenous basis, basically the here is the gene cluster. This gene cluster 1, gene cluster 2 and 3. This gene cluster contain a arginine containing gene E, arginine containing gene F, arginine containing G, and arginine gene containing E, H or F. So here that means this gene will lead to produce the messenger RNA during the transcription. So after the transcription, that messenger RNA will produce from each gene. So the each gene contain a specific function to produce a specific protein during translation. So important things is that the translation will lead to produce protein. Protein can be another type of protein, for example building block protein or enzyme protein or functional protein or structural protein, etc. But here we are taking the enzyme which that is produced from the arginine containing gene E, produce the enzyme E, while on the other hand the other gene produce the enzyme F. After the translation from these messenger RNA, each messenger RNA will transcribe from the different gene, and the different gene containing a messenger RNA will contain a translation, and that translation produce the protein. This protein is the basically the phenotype, while the genotype is the genetic makeup, while the phenotype is the physical makeup. So this is the physical makeup, basically is the enzyme E, enzyme F, and enzyme G and enzyme H. So this enzyme is collectively important for the production of arginine through the gene. So this is the 1Gene 1Polypeptide mechanism, which that means the here is the one gene clusters. Basically the one thread DNA contain a gene clusters will lead to control the messenger RNA and produce the enzyme. This enzyme is lead to produce the arginine. This arginine is used for the polypeptide chain of the amino acid, which that is arginine is important for it. So that's important thing is that in the body the glutamate is basically converted into the arginine by this enzyme activity. For example, the enzyme E will that glutamate will convert into the ornithine, and ornithine will convert into the citrulline by the enzyme F, and the citrulline will convert into the arginosuccinate by the action of the enzyme G, and the arginosuccinate will convert into the arginine, which that in the presence of enzyme H. So that means arginine is important for the polypeptide. So that means in our body the arginine is basically the non-essential amino acid which that is our body will be synthesized. So just like that, and just like a neurospora is also produced the arginine. Arginine is the amino acid which that is used for the metabolic pathway, and the polypeptide missing is the arginine, which that is basically arginine not present, so the function will not produce, growth will not produce. So here is the lack of arginine is the abnormal function. While we are giving the supplementation of arginine, so the normal function can be possible. So that means the for example the normal function can be possible due to the arginine. So the arginine will be important. So that means the 1 gene 1 polypeptide mechanism in this manner. So then 1 gene is important for the 1 polypeptide. For example, in 1 polypeptide arginine is missed, so the arginine is necessary. For example, so the sickle cell anemia or some disease can be occur due to the 1 gene changes. So that's 1 gene changes is known as a point mutation, which can be a harmful or helpful. So the without arginine the minimal media will not produce, while the minimal media contain arginine can produce. So thanks.